Imagine you've just spent the entire morning out foraging in the woods.
Right, getting your hands dirty.
Exactly. You've got a basket completely full of wild mushrooms. And, you get home, you sit down at your kitchen table, and you pull out this heavy, trusted field guide. Like maybe one that's literally been sitting on your shelf for decades.
The kind with the glossy photos and the very authoritative text.
Yeah, the definitive guide. And you start identifying your haul, right? You flip to a page and you see a picture that just perfectly matches this odd sort of saddle-shaped mushroom sitting right there in your hand.
Hmm.
And the book, I mean, it confidently tells you exactly what it is. It gives you this neat little Latin name, categorizes it perfectly. But what if I told you that eating that perfectly identified mushroom could actually flood your bloodstream with a chemical compound that is literally used by the military to launch rockets into space?
Wow. I mean, the sense of betrayal you'd feel toward that field guide would just be profound.
Right. Like you trusted it.
Exactly. Because we sort of naturally expect our reference books to be these permanent binary arbiters of truth, like safe or deadly.
Yeah, black and white.
But the thing is, when you step into the world of mycology, the study of fungi, that absolute authority, it just crumbles almost instantly. We constantly find ourselves looking at a taxonomic landscape that is, well, it's just constantly shifting right beneath our feet.
It really is like the ultimate diagnostic muddy water. And that's why today's deep dive is dedicated entirely to this one specific organism that has just thrived in those muddy waters. I mean, completely fooling scientists, seasoned foragers, toxicologists, all of them for nearly 250 years.
Which, if you think about it, is a remarkably long time for a single macroscopic organism to just hide its true identity. And, hide its true chemical nature right there in plain sight.
Yeah, it's wild. So we are tracking down the hooded false morel. Now, historically, if you open that old field guide we were just talking about, you're going to find it listed under the scientific name Gyromitra infula.
Or the classic name.
But today, because of some totally radical reclassifications in the scientific community, it's officially known as Paragyromitra infula, or you might also see it referred to by its, I guess, somewhat whimsical common name, the elfin saddle.
The elfin saddle. I always like that one. But our mission today is really to unravel the biological, the chemical, and the historical mysteries of this specific fungus.
Yeah.
Because we are pulling together centuries-old taxonomic journals, cutting-edge phylogenomic studies from 2023 and 2025. We're looking at modern toxicology reports, molecular forensic papers.
The whole 9 yards.
Exactly. We really want to understand how this one single mushroom basically forces us to question our assumptions about, well, how things are related in nature, how toxins are actually distributed, and frankly, how we identify danger out in the wild.
Okay, so to get us into the right headspace for this, I want you to just picture the environment where this mystery starts. The backdrop to our whole conversation today is this shadowy, very damp forest floor. It's late autumn.
Okay, setting the scene.
Yeah. So there's this crisp chill in the air. The leaves are mostly down, and the light filtering through the canopy is pretty weak. And right there, growing directly out of this decaying conifer log, is this large, strange, saddle-shaped mushroom.
Just sitting there on the dead wood.
Exactly. And it's this deep reddish brown, maybe pushing into like a chestnut color. And it's perched precariously on this hollow whitish stem. Like it does not look like your standard grocery store mushroom.
Right. No neat little umbrella cap.
No gills underneath. It looks jagged. It looks honestly almost alien.
Well, the morphology, and by that I mean the actual physical structure of the fruiting body, is incredibly striking. And if we want to understand why this fungus is so controversial today, we really have to look closely at that bizarre physical shape.
Because the shape is the problem.
Exactly. That shape is the very thing that led to centuries of mistaken identity.
I mean, I've seen pictures in the source material we're looking at, and honestly, it just looks like a mutated fortune cookie.
A mutated fortune cookie. That is, that visual captures the mechanical tension perfectly.
Wait, mechanical tension?
Yeah, so typically Paragyromitra infula grows to be about, say, 2.5 to 9 centimeters wide, and roughly the same in height. And the cap is that striking reddish brown, sometimes leaning into a sort of vinaceous wine-colored brown.
Okay.
But unlike true Morels, which have that pitted honeycomb sponge look, or the brain-like convolutions of the infamous false Morel Gyromitra esculenta, this elfin saddle develops a very distinct arching shape. It features these 2 to 4 sharp-tipped lobes that point upward.
Which creates that saddle look.
Exactly, creating something that looks very much like a riding saddle.
But wait, why would it even grow like that? Because a standard mushroom cap, the umbrella shape, that makes evolutionary sense. It acts like an umbrella to protect the delicate spores underneath from the rain, so they can just drop into the wind current.
Right, standard spore dispersal.
Yeah, so why does this one look like a crumpled saddle? It just seems kind of counterproductive.
It all comes down to this really fascinating developmental quirk. It's a sort of biological tension.
Okay, how so?
Well, very early in the mushroom's development, the outer edge of its cap anchors tightly to the stike.
The stipe being the stem.
Right, the stipe is the hollow, whitish stem supporting the whole structure. So the edge fuses to the stem. But the hymedium, which is the fertile spore-bearing surface that covers the outside of the cap, it doesn't stop growing. It just continues to rapidly expand and multiply its cells.
Oh, wow. So the edges are literally glued down, but the middle is just still inflating.
Exactly. Imagine taking a piece of fabric, pinning all 4 corners firmly to a small wooden board, and then somehow forcing the fabric to double its surface area.
It's got to bunch up.
The fabric has nowhere to go. It buckles under the pressure of its own expansion, it folds, it arches upward, and it just crumples into itself, creating those sharp, angular lobes. So the physical form of the elfin saddle is dictated almost entirely by this mechanical tension, rather than some master blueprint for a specific aerodynamic shape.
I love that. Like trying to inflate a balloon inside a small box so it just kind of crumples. And, that tension is a really great metaphor for its history in the scientific literature, too.
It really is.
Because early mycologists looked at that folded, crumpled shape, and they made some massive assumptions that locked this mushroom into the completely wrong category for generations. So let's trace this taxonomy nightmare. Where does the paper trail for this thing actually begin?
Well, we have to travel way back to 1774. A German polymath and mycologist named Jacob Christian Scheffer was actually the first to officially describe this fungus in the scientific records.
1774, that is a long time ago.
It is. He was cataloging the fungi of Bavaria, and he grouped this specimen with other saddle-shaped mushrooms he had found. And he named it Helvella infula.
I was actually looking at the etymology of that specific epithet, infula, and it is fascinating. It comes from a Latin word referring to this heavy, twisted wool band that Roman priests wore around their heads during religious sacrifices.
Oh, that's interesting.
Right. You look at the twisted, folding lobes of the mushroom, and you can see exactly why Schaefer chose that specific word.
Yeah, it was a highly descriptive name. And you have to remember, descriptive naming was the absolute gold standard for taxonomy in the 18th century. Because, without advanced microscopes, without genetic testing, scientists categorized the natural world entirely by macromorphology.
Just by what they could see.
Exactly. If things look similar to the naked eye, they were placed in the same family folder.
Makes sense for the time.
Right. But taxonomy is a living discipline. It's constantly undergoing revision. So by 1849, the eminent Swedish mycologist Elias Magnus Fries looked at the broader landscape of fungi and decided that certain mushrooms need their own distinct genus based on their gyro surfaces.
Gyros meaning like deeply convoluted or brain-like.
Precisely. And he created the genus Gyromitra.
And because our little elfin saddle had all those crumpled folds and lobes, it was inevitably going to get swept into that new folder.
It was unavoidable. In 1886, the French mycologist Lucien Quelet did exactly that. He took Scheffer's twisted little elfin saddle, noted its convoluted surface, and officially moved it into Fries's new genus, calling it Gyromitra infula.
And there it sat?
And there it sat. For over 130 years, if you found an elfin saddle in the woods, the entire scientific consensus told you to call it a Gyromitra. It was considered a close blood relative to the highly toxic brain mushroom, Gyromitra esculenta.
Just because they both looked kind of wrinkly and brainy and they grew in roughly similar ways. I mean, it's wild to think about how much weight was placed on purely visual evidence back then.
It was all they had.
True. Which brings us crashing into the 2023 and 2025 phylogenomic bombshells. We finally reached a point in science where we didn't have to rely on what a mushroom looked like to the naked eye. We could actually read its genetic code.
The ultimate truth serum.
Exactly. But looking at the sources, the scale of this study is just staggering. They didn't just sequence a couple of genes here. They sequenced 75 whole genomes from the family Dissinaceae. And they analyzed something called, and I'm going to read this directly, 1,542 concatenated Busco genes.
It's a mouthful.
It is. I have to be honest, concatenated Busco genes sounds like an impenetrable wall of jargon. Help us break that down. What does that actually mean for the rest of us?
Let's absolutely strip away the jargon because the
Yeah.
underlying concept here is a brilliant tool for understanding evolution. So BUSCO stands for Benchmarking Universal Single Copy Orthologs.
Okay, still sounds like jargon.
Fair enough.
Yeah.
You can just think of them as core essential genes. These are pieces of DNA that are so fundamentally important to basic cellular function that they are universally shared across a vast range of species.
Okay, got it.
And importantly, they usually only occur once in an organism's entire genome. They don't duplicate and muddy the waters.
So they're like the foundational load-bearing pillars of the genetic code. Like every single organism in this group has to have them just to survive.
Exactly. Think of it like comparing 75 different encyclopedias. If you just grab a random page from each book, you might be comparing an entry on apples in one book to an entry on zebras in another.
Which tells you basically nothing.
Right. It tells you nothing about how the books actually relate to each other. But BSCO genes are like identifying 1,542 specific chapters that appear in every single encyclopedia, say, the chapter on photosynthesis or the chapter on cell division.
Oh, I see.
And by concatenating them, scientists just string those 1,542 specific chapters together in a row. They can then go through, letter by letter, and compare the exact sequence across all 75 mushroom species.
Okay, that makes so much sense. It provides this hyper-accurate, high-resolution map of their evolutionary family tree. It's essentially a massive 23andMe test, but instead of telling you that you're, I don't know, 5% Neanderthal, it's telling us exactly when these mushrooms diverged from a common ancestor millions of years ago.
That's a perfect analogy.
So what did this massive genetic map actually reveal about our elfin saddle?
Well, the revelation was absolutely shocking to classical mycologists. It proved definitively that the physical shape of a mushroom is a terrible, highly deceptive indicator of its evolutionary lineage.
Take that, visual taxonomy.
Seriously. The family Dicinaceae had to be split into two entirely different tribes, Dicinia and Gyromitri, and Gyromitra infula. It was completely ripped out of the genus it had belonged to since 1886. It was evicted.
Evicted and given a brand new name, Paragyromitra infula. The prefix para meaning like beside or closely related to, but distinctly separate from the true Gyromitra.
Exactly. But the real shock, the thing that really threw people, came when they traced the newly named Paragyromitra line back to its closest evolutionary relative.
Okay, lay it on me.
Well, when you look at an elfin saddle, this above-ground saddle-shaped mushroom perched up on a white stem, you'd naturally assume its closest cousin is another mushroom that looks roughly like that.
Right. Yeah, something with a stem and a cap.
But the genetic map revealed its closest relatives actually belong to the Hydnotria genus.
Okay, I looked up Hydnotria in the research, and I honestly could not believe the images. They are truffles.
Yes.
They are completely subterranean, stemless, potato-like lumps that grow entirely underground. How is that possible?
I know, it's wild. The genetic link between an above-ground saddle mushroom and a subterranean truffle just completely shatters the classical idea that macroscopic physical similarity equals biological
This
relation.
is mind-blowing.
And the implications of this are massive.
Yeah.
It tells us that above-ground shapes like saddles or caps or brains
Right.
are just evolutionary elaborations. They're highly plastic structural adaptations of a common ancestor.
Oh, wow.
Right, so that ancient ancestor had descendants that kept reaching for the surface, utilizing that mechanical tension to fold and drop spores into the wind.
Yeah.
But other descendants of that exact same ancestor abandoned the surface entirely. They lost their stems, they lost their caps, and they basically learned to live completely underground, relying on animals to dig them up and spread their spores.
That is just incredible. So a mushroom that grows on a decaying log and looks like a folded saddle is actually genetically closer to an underground, dirt-covered truffle than it is to the brain mushroom grown just a few feet away from it on the forest floor.
That's exactly what the genes tell us.
Yeah.
And honestly, it forces us to completely reevaluate the ecological behavior of this fungus. Because it is genetically distinct from the other false morels it was historically lumped in with, we really shouldn't be surprised that its lifestyle, its phenology, and ecology is completely different too.
Right. And let's get into that lifestyle, specifically what I kind of call the autumnal paradox. Because when most people think of foraging for morels or even false morels, they immediately think of spring.
Definitely. It's a huge spring event.
Yeah, it's a deeply ingrained cultural tradition in a lot of places. You
Yeah.
know, the snow is melting, the ground is thawing, the first green shoots are coming up, and people head into the woods to find these mushrooms. But Paragyromitra infula missed that memo entirely.
It is a stark outlier in its family. While its distant cousins are popping up as companions to the spring thaw, P. infula fruits almost exclusively in late summer and autumn.
It's like the goth cousin of the mushroom family.
Basically, if we look at granular observation data from the Pacific Northwest, for example, nearly 50% of all sightings of this fungus occur in November.
November. I mean, in the Pacific Northwest, November is incredibly cold. It's dark, it's wet, it's practically winter. Why does it wait so long?
Well, it requires very specific ecological triggers that simply do not exist in the spring. It waits for the sustained heavy moisture of the fall rains to fully saturate its environment. And crucially, it requires the soil temperatures to drop significantly, usually well below 45 degrees Fahrenheit. It thrives in the chilling damp.
And its diet is totally different, too. It is a saprobe.
Yes, and to appreciate that, consider how many of our famous, highly prized edible mushrooms function. Truffles, chanterelles, porcinus, they often form mycorrhizal partnerships.
Meaning they work with living trees.
Right. They wrap their mycelium around the living roots of trees, creating this symbiotic exchange where they trade minerals from the soil for sugars produced by the tree's canopy. They rely on a living host. But Paragyromitra infula is a saprobe. Its primary ecological function in the forest is decay. It eats the dead.
It breaks down rotting conifer wood, like Douglas fir, spruce, pine.
Exactly. It targets the complex lignins and celluloses in dead wood, breaking them down and returning those locked-up nutrients back to the soil. You might find an elfin saddle growing conspicuously on the side of a massive, decaying stump.
Right.
But sometimes you'll see it growing straight out of what looks like hard-packed dirt on a forest trail. However, if you carefully dig down beneath the stem, you will almost always find a piece of buried, rotting wood that the mycelial network is anchored to and feeding on.
And this specific lifestyle, eating dead conifer wood in near-freezing, damp, late autumn conditions, it actually sets the stage for something incredible. Because of this exact combination of factors, this fungus is the engine behind one of the most magical, mysterious physical phenomena you can witness in the forest.
The formation of hair ice.
Yes.
Oh.
Hair eyes, as it was originally documented in German. Picture this. You're walking through that freezing, damp November forest we talked about. You look down at a dead log lying on the ground, and it appears to have sprouted this luxurious, flowing mane of pure white, silky hair.
It's a crazy visual.
It looks almost like a small white animal is resting on the wood, or like, I don't know, synthetic cotton candy.
Yeah.
The strands are incredibly fine, and they wave and curl together. But if you reach out and touch it, it instantly melts against the warmth of your skin, because it's not hair, it's solid ice.
It is a breathtaking biophysical phenomenon, and for a long time, it was deeply, deeply misunderstood.
Oh.
Yeah. Scientists knew that hair ice forms on moist, decaying wood in very specific meteorological conditions, specifically high humidity combined with sub-zero temperatures, usually hovering right around -4 degrees Celsius.
Okay, freezing but not crazy cold.
Right. But there was a missing variable, because you can take a piece of dead wood, put it in a highly advanced climate-controlled chamber at exactly -4 degrees with perfect humidity, and hair ice will never, ever form.
Unless a very specific organism is living inside that wood.
Exactly. Researchers finally discovered that the presence of specific living fungi is the mandatory catalyst, and Paragyromitra species are among them, along with a few others, most notably a crust fungus called Exidiopsis effusa.
But how does a fungus create hair made of ice? I mean, it sounds like magic, but obviously it's physics. Walk us through the biophysics of this. What is the mushroom actually doing inside that log?
We have to look at the metabolic rate of these specific fungi. Most organisms, including many other fungi, go dormant as the temperature approaches freezing. Their metabolism just slows down to conserve energy.
Like hibernation.
Exactly. But Paragyromitra and Exidiopsis remain metabolically active even as the wood around them drops below zero. They continue to aggressively digest the dead wood. And as they break down those complex carbohydrates, their cellular respiration produces two key byproducts: heat and gases, primarily carbon dioxide.
Okay, so the fungus is essentially breathing and generating this tiny amount of warmth inside the decaying log.
Yes, and that metabolic activity creates a localized zone of internal hydrostatic pressure within the wood. Now, to understand the ice, we have to look at the anatomy of the wood itself. Hardwoods and conifers have these microscopic radial channels called medullary rays, which originally transported nutrients when the tree was alive.
Oh, like tiny little pipes.
Exactly like tiny pipes. So the internal pressure created by the fungal gas pushes the supercooled liquid moisture inside the wood outward, forcing it through these microscopic pores at the surface.
How microscopic are we talking here?
Incredibly tiny. These pores are often a single cell wide, perhaps a fraction of a millimeter in diameter.
Wow. Okay, so you have this fungal pressure cooker inside the wood, constantly pushing super cooled water toward the surface. It's basically like a microscopic Play-Doh fun factory. The water gets squeezed out through these tiny cellular stencils.
That analogy perfectly illustrates the mechanical extrusion process. As that infinitesimally thin string of supercooled liquid water breaches the surface and hits the sub-zero atmospheric air, it instantly nucleates and crystallizes into solid ice.
Instant freeze.
But the real magic is in the continuous pressure. Because the moisture is continually being pushed out from behind by the fungal metabolism, the ice doesn't just form a flat crust, it grows outward from the base. The new ice pushes the old ice forward, forming these long, continuous, incredibly fine filaments that can grow over 5 centimeters long in a single night.
That is amazing. So the individual strands of ice are actually shaped by the shape of the microscopic pores in the wood, and they're powered by the breath of the fungus. The mushroom isn't just sitting there passively. Its basic survival metabolism is actively engineering its physical environment. It's manipulating local thermodynamics to extrude these beautiful ice sculptures.
It is a profound example of biology driving localized physical phenomena. But, as beautiful and delicate as hair ice is, the organism that helps create it, our elfin saddle, has carried a very dark, terrifying reputation.
Yeah, the dark side of the mushroom.
Despite its role in forest magic, Paragyromitra infula has been universally feared for over a century.
Which brings us to the most gripping part of the story, the rocket fuel myth.
Oh, yes.
For over 100 years, this exact mushroom, the one creating the delicate hair ice, was believed to be packed full of a chemical compound that aerospace engineers literally used to launch rockets into space. But as we dig into the research, we find that the chemistry of this organism was just as tragically misunderstood as its taxonomy.
The scientific literature from the 20th century is just filled with dire warnings about this fungus. And those warnings were built around a highly specific, very dangerous chemical.
Okay, so I was looking through the toxicology notes on this, and the chemical name of the primary toxin is, all right, let me try this, acetaldehyde N-methyl N-formal hydrazone. I'm definitely butchering that pronunciation.
No, you're close enough. Acetaldehyde N-methyl N-formal hydrazone. But honestly, scientists and toxicologists refer to it by its much simpler, infinitely more infamous common name, gyromitrin.
Gyromitrin. Named after the Gyromitra genus, naturally.
Exactly. In fresh, toxic Gyromitra mushrooms, like the closely related brain mushroom, Gyromitra esculenta, you can find staggering amounts of this compound. We are talking anywhere from 50 to 300 milligrams of gyromitrin per kilogram of fresh mushroom tissue.
That sounds like a lot.
It is. It's a highly volatile, highly water-soluble mycotoxin. And the way it kills a human being is exceptionally grim. To understand it, we really have to look at toxicokinetics.
Toxicokinetics. Break that down for us.
It's the study of how a chemical substance enters the body, how the body alters it, and the specific mechanisms by which it causes damage.
Okay, so walk us through this poisoning cascade. Let's say a forager in the 1970s looks at their field guide, misidentifies a toxic false morel, cooks it up, and eats a meal loaded with gyromitrin. What physically happens as that mushroom travels down their digestive tract?
Well, the danger doesn't actually begin until the gyromitrin reaches the stomach. In its raw form, gyromitrin is relatively stable. But when it enters the human stomach, it encounters a highly acidic environment.
Stomach acid.
Right. And gastric acid triggers a violent chemical reaction called acid hydrolysis. The acid effectively cleaves the gyromitrin molecule apart, stripping away certain chemical groups and converting the parent compound into a totally new, highly reactive chemical.
Monomethylhydrazine.
Monomethylhydrazine, or MMH. Wait, isn't that the rocket fuel?
Literally. Monomethylhydrazine is a highly toxic carcinogenic hydrazine derivative. It is currently used as a hypergolic bipropellant in aerospace engineering. Hypergolic meaning it ignites on contact with another chemical.
Exactly. NASA and the military use it in the orbital maneuvering systems of spacecraft. It is incredibly volatile and reactive. It boils at just 87.5 degrees Celsius, which is significantly lower than the boiling point of water.
I am still trying to wrap my head around this. You're telling me a mushroom sitting in the dirt naturally synthesizes a precursor compound that, when mixed with human stomach acid, turns into the exact same chemical NASA uses to propel satellites in a vacuum. How does that even happen? It feels like science fiction.
It really highlights the astonishing biosynthetic capabilities of fungi. They are essentially master chemists, assembling complex molecules from basic environmental building blocks for defense or metabolic regulation. Unfortunately for us, the specific molecule they build happens to be devastatingly toxic to mammalian biology.
Yeah, that's an understatement. So you have actual rocket fuel forming in your stomach acid. What does MMH do once it crosses the stomach lining and gets into your bloodstream? How does it start dismantling the body?
Once MMH enters the systemic circulation, it aggressively targets the central nervous system. Specifically, it hunts for a highly specific enzyme called pyridoxal phosphokinase.
Okay.
The MMH molecule binds tightly to this enzyme, completely inhibiting its function. Now, why does that matter? Because pyridoxal phosphokinase is the enzyme responsible for activating vitamin B6, which is also known as pyridoxine.
So the rocket fuel chemically handcuffs the enzyme, preventing your body from using vitamin B6. But what does vitamin B6 do that makes its absence so lethal?
Vitamin B6 is an essential cofactor. Think of it as the key that turns the ignition for synthesizing a critical neurotransmitter called GABA, gamma-aminobutyric acid.
Okay, I recognize GABA from neurobiology. If we translate this into everyday mechanical terms, GABA is essentially the brain's primary brake pedal.
Right.
It is the main inhibitory neurotransmitter. Its job is to calm the nervous system down, to counteract excitatory signals, and to prevent your neurons from firing too fast or uncontrollably.
That is an excellent way to conceptualize it. It is the regulatory dampener for your entire nervous system. So trace the cascade. The MMH blocks the enzyme can't activate vitamin B6. Without B6, you cannot synthesize GABA, and without GABA.
The brakes are cut. The brain loses its ability to slow down electrical signals. The nervous system just revs completely out of control.
Exactly. And that catastrophic biological failure leads to what toxicologists call Gyromitra syndrome. And one of the most insidious aspects of this syndrome is the delayed onset.
Because it takes time for the body to process it.
Right. A person eats the meal and feels perfectly fine for hours. The hydrolysis and the enzymatic inhibition take time. About 5 to 8 hours post-ingestion, the patient typically experiences severe, sudden gastrointestinal distress, violent nausea, vomiting, abdominal cramping.
That sounds awful enough.
It gets worse, because the true horror begins as the GABA depletion reaches a critical threshold in the brain.
Because the brakes are completely gone, what happens to the patient then?
This leads to a state called epileptogenic neurotoxicity. The uncontrolled firing of neurons manifests as severe, intractable seizures. The patient often suffers from intense delirium, loss of motor control, and eventually a descent into a deep coma. The neurological storm is just devastating.
And if the brain damage wasn't terrifying enough, the sources indicate that MMH attacks the body on multiple fronts simultaneously.
Yes, MMH is also highly hepatotoxic and causes severe oxidative stress to cellular structures. It triggers acute hemolysis.
Hemolysis. Hemo meaning blood. Alesis meaning to break or burst. Why does it burst red blood cells?
The MMH compound damages the fragile lipid bilayers, the outer skin of the red blood cells. The cellular membrane weakens and ruptures, spilling hemoglobin into the bloodstream.
Which is really bad for your organs.
Very bad. This massive release of cellular debris can quickly overwhelm the kidneys. Simultaneously, the toxin causes profound liver necrosis. It literally murders the tissue of the liver. The combination of intractable seizures, exploding red blood cells, and acute liver failure makes gyromitrin poisoning an exceptionally terrifying and difficult condition to treat.
And for a century, every field guide, every mycologist, every toxicologist pointed their finger at our little autumn fortune cookie, Paragyromitra infula, and said it contained this exact poison. They labeled it a deadly biohazard.
They did.
But then we arrive at the 2023 paradigm shift.
This is where modern analytical chemistry completely rewrites the narrative. A groundbreaking study in 2023 decided to put the elephant saddle to the ultimate test using a technology called ultra-high-performance liquid chromatography coupled with mass spectrometry.
Ultra-high-performance liquid chromatography. How does that technology even work? Is it just like a highly advanced chemical sieve?
Essentially, yes. It is an incredibly sensitive analytical technique. You take a sample of the mushroom, extract the chemical compounds into a liquid solvent, and force that liquid through a column packed with specialized microscopic particles at extremely high pressure.
Okay, forcing liquid through a tube.
Right, and different chemicals travel through the column at different speeds based on their molecular structure. As they exit the column, a mass spectrometer literally weighs the individual molecules. It allows scientists to separate, identify, and measure the tiniest, almost imperceptible chemical components of a sample.
Wow.
So they analyzed verified, genetically confirmed specimens of Paragyromitra infula, specifically hunting for any trace of gyromitrin.
And what did the mass spectrometer find?
Zero. Absolutely zero detectable gyromitrin. Not a trace.
Zero. I just find that staggering. For over 100 years, this specific mushroom was labeled a deadly rocket fuel-laced biohazard. People feared it. It was listed as toxic in every major text. How could the entire scientific community get something so profoundly wrong for so long?
The answer lies in the profound danger of visual identification in mycology. It circles back to those diagnostic muddy waters we discussed at the very beginning.
The lookalikes.
Exactly. Paragyromitra infula is almost visually identical to another species that shares its environment, a species called Paragyromitra ambigua. And Paragyromitra ambigua definitively does contain toxic levels of these hydrazine compounds.
So early foragers and scientists were eating ambigua, getting horribly sick, suffering from liver failure or dying, and the medical community was pointing at infula and taking its mugshot. It was a tragic case of mistaken identity. But how do you even tell them apart if they look identical in the field?
To the naked eye of a forager standing in the woods, it is nearly impossible. P. ambigua sometimes exhibits a slightly more irregular wavy cap, and its coloration can occasionally skew a bit more violet. But these traits exist on a highly variable spectrum influenced by local moisture and sunlight.
So you can't rely on that at all.
No, they are not reliable diagnostic markers. The only definitive, absolute way to tell the safe elfin saddle from his deadly twin is under a high-powered microscope. And the physical difference is chillingly small.
We are talking about differences at the cellular level here.
We are talking about the spores themselves. The microscopic seeds the mushroom drops. If you place the spurs of the non-toxic P. infula under a microscope at massive magnification, you will see that the ends of the oval-shaped spores are perfectly smooth.
Okay, smooth, rounded ends. And the deadly ambiguous spores.
The spores of the toxic P. ambigua possess what mycologists call blunt apiculi. They have these tiny, distinct bumps on the far ends of the oval. And when I say tiny, I mean they measure about 1.5 to 3.0 micrometers in length.
Wait, let me contextualize that. A micrometer is 1/1,000,000th of a meter. A human red blood cell is about 8 micrometers across. So you're saying that a tiny structural bump that is smaller than a single human blood cell is the only physical difference between a safe, harmless mushroom and a catastrophic trip to the emergency room with rocket fuel poisoning.
That tiny microscopic bump is the razor-thin line between safety and hepatotoxicity, which perfectly explains why the error persisted for an entire century. Very few amateur foragers, and honestly very few early field mycologists, were bringing oil immersion microscopes into the woods to measure spore apiculi before making their field classifications or writing their field guides.
Yeah, nobody's doing that on a hike.
Exactly. They saw a saddle shape, assumed it was infula, got poisoned, and the legend grew.
Okay, but I have to push back here for a second. I am looking at the modern foraging advice in our sources. If the 2023 chromatography study proves definitively that true P. infula has absolutely zero gyromitrin, why are all the contemporary experts and foraging associations still screaming at us to never, ever eat it? If the poison isn't there, shouldn't it be officially cleared of all charges and put on the edible list?
That is a highly logical question, but we really have to approach foraging from a perspective of extreme pragmatism and risk management. There are 3 critical reasons why P. infula is still universally listed under the bold heading of "do not eat."
Okay, what are they?
First, and most obviously, as we just discussed, the risk of misidentifying it with the deadly P. ambigua is simply too high. Unless you are carrying a compound microscope and a glass slide into the forest, you cannot be certain which species you are holding in your hand. The cost of being wrong is intractable seizures and liver necrosis.
Fair enough. Playing a game of Russian roulette where the bullets are microscopic bumps is a terrible hobby.
Exactly. The second reason is purely culinary. Even if you knew with absolute genetic certainty that you were holding a safe infula, the flesh of the elfin saddle is incredibly thin, papery, and brittle. It has almost no mass or texture. It lacks the dense, meaty substance that makes a true morel so prized in French cooking.
So it's not even worth the risk. It'd be like risking your life to eat a piece of unflavored, wet cardboard.
Precisely. It is gastronomically useless. And the third reason, which is arguably the most important from a scientific standpoint, is that just because a mushroom lacks gyromitrin does not mean it is safe.
Oh, right. There could be other stuff.
Exactly. The absence of one specific famous poison does not equate to the presence of safety.
Hmm.
Fungi are complex biochemical factories. There is a high probability that there are other secondary metabolites, other toxic compounds in P. infula that we simply haven't isolated or identified yet that could cause profound gastrointestinal distress or subtle liver damage over time. We simply don't know enough to declare it safe.
Okay, so the visual difference between the non-toxic elephant saddle and its deadly cousins is practically non-existent. And because people, either out of sheer ignorance, extreme risk tolerance, or starvation, do still mistakenly forage and eat these saddle mushrooms, the medical community faced a massive crisis.
A huge crisis.
Imagine a patient arriving at a rural emergency room suffering from acute seizures and vomiting. The doctors urgently need to know exactly what species of mushroom they ate to administer the correct targeted therapy. But you can't identify a mushroom shape or look for microscopic spore bumps after the mushroom has been chopped up, heavily boiled in a stew, and partially digested in a bath of human stomach acid.
This is the critical bottleneck in treating toxicological emergencies. When the physical evidence is reduced to a pile of unrecognizable, semi-digested brown mush, traditional morphological identification fails completely. You cannot use a field guide on stomach contents.
Which brings us to the marvel of modern molecular forensics. The doctors needed a way to identify the ghost of the mushroom from its surviving DNA. But why couldn't they just use standard genetic testing? We hear about PCR testing all the time, especially after the pandemic. Why not just run a PCR swab on the stomach contents?
Well, traditional genetic testing methods like standard PCR polymerase chain reaction or the LCMS chemical analysis we discussed earlier are incredibly accurate. They are the gold standards of laboratory science. But they face severe limitations in the real world of emergency medicine.
Like what?
They are expensive, they are slow, and crucially, they require giant, complex, highly sensitive laboratory machines.
Right. From what I understand, standard PCR requires a process called thermal cycling. You have to put the sample in a machine that heats it to almost boiling around 95 degrees Celsius to physically melt the double helix of the DNA apart. Then the machine has to rapidly cool down to let the targeted primers attach to the sequence and then heat up again to build the new strand. And it just repeats this hot, cold, hot cycle dozens of times.
And that precise thermal cycling requires a specialized, heavy, expensive machine. That is not a useful solution in resource-poor environments. Consider where many of these poisonings occur. They frequently happen in rural, remote areas such as the mountainous villages of rural China, where foraging is a common part of subsistence living.
Right, they don't have massive labs.
Exactly. A small village clinic or a remote county hospital does not have a $100,000 mass spectrometer, nor do they have a delicate PCR thermal cycler.
And more importantly, they don't have the luxury of time. When a patient is actively seizing from GABA depletion and suffering from acute hepatotoxicity, you don't have 3 days to carefully package a biological sample and mail it to a centralized metropolitan laboratory for sequencing. You need a definitive answer in an hour so you can start administering massive intravenous doses of vitamin B6 to stop the seizures.
This urgent life-or-death pressing need led molecular biologists to develop a brilliantly elegant, robust diagnostic tool known as LAMP.
LMEP, loop-mediated isothermal amplification. I dug into the papers on how this works, and it honestly feels like science fiction compared to the older methods. Let's break down the mechanics of LAMP step by step.
It represents a massive paradigm shift in point-of-care diagnostics. First, LAMP needs a target. It targets a very specific region of the fungus's DNA called the RTVE, or internal transcribed spacer region. This is a segment of non-coding DNA situated between the genes that produce ribosomal RNA.
Wait, why target the spacer region instead of an actual active gene?
Because the ITS region is evolutionary gold for identification. Active genes, like those required for basic metabolism, are highly conserved. They look very similar across many different species. But the spacer regions mutate rapidly. They're highly conserved within a single species, but they vary wildly between different species.
Oh, I get it.
This makes the ITS region the perfect, highly specific genetic barcode for identifying exact mushroom species.
But how does the LMP test guarantee it's finding the exact right barcode for our specific elfin saddle and not accidentally grabbing the DNA of the safe button mushrooms the patient ate in the same stew?
Through a system of extreme redundant precision. While a standard PCR test might use just two primers, small pieces of custom DNA designed to find it attached to the ends of the target sequence, the LMP assay designed specifically for P. infula uses a complex set of 6 specific binding regions targeted by 4 to 6 distinct primers.
6 different primers all hunting for the same target.
Exactly. These primers are designed to recognize 6 completely distinct, separate sequences along the ITS barcode. For the amplification reaction to proceed, all 6 of those sequences must perfectly match the target DNA.
And if they don't?
If even one is off, the reaction stalls. The mathematical improbability of another organism's DNA accidentally matching all 6 binding sites practically eliminates the risk of false positives.
That is incredibly smart. That handles the accuracy. But the real game changer for those rural clinics is the isothermal part of loop-mediated isothermal amplification, right?
Absolutely. Isothermal simply means it operates at a single, constant temperature throughout the entire process. There is no complex mechanical heating and cooling cycle required to manipulate the DNA. You can run the entire reaction at a steady, unwavering 65 degrees Celsius.
Which means you don't need the expensive thermal cycler machine. You just need a way to keep a small tube warm, a basic thermostatic water bath. You could theoretically run this test using a digitally controlled pot of warm water.
That is the beauty of it. But how does it unzip the DNA without the high heat used in PCR? The elegance lies in a special enzyme required for the reaction, a strand displacement DNA polymerase. This enzyme is usually derived from a heat-loving bacterium called Bacillus staurothemophilus, often abbreviated as BLyst.
What makes this BLyst enzyme so special?
Standard polymerases need the DNA double helix to be melted apart into 2 single strands before they can start reading and copying. The BLyst polymerase doesn't need that. It possesses strand displacement activity.
Meaning it forces its own way in.
Think of the DNA double helix like a tightly zipped zipper. Standard PCR uses extreme heat to completely melt the zipper apart into 2 separate halves. The BLyst enzyme works differently. It acts like a wedge or a microscopic snowplow.
A snowplow.
Yeah. Once the primer attaches, the BLyst enzyme starts building the new DNA strand, and as it moves forward, it physically plows through the double helix, unzipping and pushing the old complementary strand out of the way as it goes. It does the mechanical work of separating the strands without needing thermal melting.
Okay, so a doctor in a rural clinic takes a sample of the mushy digested stomach contents. They put a tiny drop of it into a test tube containing this blessed snowplow enzyme and the 6 highly specific primers. They drop the tube into a 65-degree water bath. But how do they know if the test is positive? If they don't have fancy lab equipment, they don't have a computer to read the fluorescent outputs of the DNA replication. Do they just guess?
No, and this is perhaps the most brilliant, user-friendly aspect of the entire LMNP system for on-site diagnostics. The researchers engineered the reaction to utilize a simple, visual pH indicator dye called phenol red.
Phenol red, like the drops you use to test the pH balance of a swimming pool.
The exact same principle. We have to look at the chemistry of DNA synthesis. As the target DNA amplifies, meaning millions upon millions of copies are being rapidly built inside that tiny tube, the chemical process of grabbing free nucleotides and attaching them to the growing DNA chain releases byproducts. Specifically, every time a nucleotide is added, a pyrophosphate molecule and a hydrogen proton are released into the solution.
And releasing protons means the chemistry of the solution is fundamentally changing. It is becoming more acidic. The overall pH of the liquid inside the tube is steadily dropping as more DNA is built.
Precisely. And phenol red is a highly sensitive chemical dye that changes color drastically based on the pH level of its environment. At the start of the reaction, in a slightly alkaline state, the liquid in the test tube is a bright, clear pink. If the specific P. infula target DNA is present in the stomach contents, the massive geometric amplification of that DNA releases a flood of protons.
And the color changes.
The pH plummets, and within 60 to 90 minutes, the liquid in the tube visually changes color from bright pink to a stark, undeniable yellow.
That is incredible. So it is essentially functioning exactly like a rapid COVID test or a home pregnancy test, but designed specifically for identifying rocket fuel mushrooms.
That's a great way to put it.
A doctor pulls the tube out of the warm water bath. If the liquid is still pink, the patient is clear that specific DNA wasn't in their stomach. But if the liquid is turned yellow, The doctor knows with absolute certainty that the patient ingested a Gyromitra or Paragyromitra species, and they are in serious, immediate medical danger.
The sheer visual simplicity of reading a color change is its greatest strength in an emergency setting, and the sensitivity of this assay is staggering. The researchers didn't just test this on pristine mushroom samples in a sterile lab. They subjected the LMP assay to rigorous, simulated real-world conditions to prove its robustness.
I read about this in the methodology section. They simulated human digestion using artificial gastric juice, mimicking the highly acidic environment of the stomach. They heavily boiled the mushroom samples for extended periods to simulate cooking a stew, which severely degrades DNA. Just how sensitive did the test remain after all that abuse?
It maintained a detection limit of an astonishing 1 nanogram of genomic DNA per microliter.
Let me try to visualize a nanogram. A paper clip weighs about 1 gram. A nanogram is 1/1000th of that paperclip.
It is an unimaginably small amount of genetic material. To put that sensitivity into a practical perspective, the researchers created mixed, contaminated samples. Imagine a massive pot of stew where someone threw in dozens of perfectly safe, edible mushrooms and accidentally tossed in just one tiny fragmented piece of the toxic lookalike.
Okay.
The LAMP test successfully, consistently detected the target DNA and turned the tube yellow, even when the target mushroom made up only 1% of the total biological sample.
That is just unbelievable. A 1% fragment of a heavily boiled, acid-digested mushroom sitting in a chaotic pool of stomach contents, and the chemical snowplow still finds it, amplifies it, and turns the tube yellow. That technology actively saves lives. It gives an exhausted doctor in a remote clinic an immediate, definitive, actionable answer so they can instantly start administering the exact antidotes to halt the neurological damage before the patient slips into an irreversible coma.
It perfectly bridges the vast gap between high-level, abstract molecular biology and gritty, on-the-ground clinical necessity. It proves that our deep, seemingly academic dives into the taxonomy and genetics of these fungi aren't just exercises in categorization. Understanding the exact DNA sequence of a weird, saddle-shaped mushroom has profound real-world life-or-death applications.
Wow. Okay, let's take a breath and pull all of these incredible threads together. We have taken an absolutely wild journey today. We started by looking at a mushroom that looks like a crumpled fortune cookie, an organism that was completely, fundamentally misunderstood by the greatest scientific minds for 2.5 centuries.
A true shapeshifter in the literature.
Right. We watched it get genetically evicted from its historical genus and unexpectedly placed on a family tree right next to completely subterranean truffles. We learned how it acts as an autumn saprobe, feeding on the dead and breathing out beautiful, microscopic strings of ice in the freezing forest.
We deconstructed its darkest, most terrifying myth, clearing its specific name of the rocket fuel murder charges that had defined its reputation for 100 years, while still acknowledging the terrifyingly small, microscopic razor's edge that separates it from its deadly toxic twins.
And finally, we marveled at the sheer ingenuity of molecular forensics, the pink to yellow NMP test that utilizes bacterial snowplows and pH dyes to track this elusive fungus down even after it has been boiled, chewed, and digested in a human stomach.
If we pull back and connect this specific story to the much larger picture of scientific inquiry, the saga of Paragyromitra infula serves as a profound, humbling reminder. Science is not a static book of permanent facts. It is not the rigid, unchanging encyclopedia we sometimes wish it to be when we want simple answers.
Oh, not at all.
It is a living, breathing, constantly evolving process of continuous refinement. The old, trusted field guides sitting on your shelf right now might contain deeply held facts that will be entirely overturned tomorrow morning by a new genetic sequencing tool or a more sensitive chromatography method.
Yeah, that absolute comforting authority we crave when we open a book just doesn't exist in the natural world. Nature is messy, it's plastic, and it is constantly defying our attempts to put it into neat little boxes.
And because the natural world is so complex and messy, critical thinking, a willingness to question historical assumptions, and a commitment to continuous learning are the most powerful tools you can possess in an information-heavy world. What we know today is never the final word. It is simply the very best approximation of the truth we can achieve with the diagnostic tools we currently possess.
That is a fantastic perspective. It really is an ongoing, evolving dialogue with nature. But before we wrap up this deep dive and send you back out into the world, I want to leave you, the listener, with one final, deeply mind-bending evolutionary question to chew on.
The genetic mapping of this family raises an incredibly important, almost unsettling question about the nature and origin of the toxins themselves.
Exactly, because when those scientists mapped out those 75 genomes using the BSCO genes, they discovered something truly bizarre about the gyromitrin toxin. The genetic instructions, the actual DNA code required to synthesize that deadly rocket fuel, are discontinuously distributed across the entire fungal family tree.
It's patchy.
Very. Our elfin saddle, Paragyromitra infula, doesn't have the code, but its very close neighbors do. And more confusingly, distantly related genera, sitting on completely different branches of the evolutionary tree, also possess the exact same code to make the toxin.
Which presents a massive evolutionary puzzle. Genetic traits usually follow a clear, linear path of inheritance.
Right. So scientists are staring at this map trying to figure out what happened. Did the ancient common ancestor of all these mushrooms possess this toxic weapon? And over millions of years of evolution, did our elfin saddles simply lose the code, abandoning the weapon to save metabolic energy?
Or, and this is where the biology gets truly wild, we have to consider the mechanism of horizontal gene transfer.
Horizontal gene transfer. The idea that genetic code doesn't just move vertically from parent to child, but horizontally between completely different species living next to each other. Is it possible that a completely non-toxic mushroom could actually absorb the raw genetic instructions to manufacture rocket fuel directly from the soil, picking up a deadly, complex chemical weapon from the decaying environment around it?
It is a staggering thought. It suggests that the damp soil of the forest floor is not just a passive bed of dirt, but an invisible, actively shifting battlefield of chemical warfare, where the blueprints for the weapons themselves can literally jump across species boundaries in the dark.
So the next time you find yourself walking through the autumn woods, and you spot a strange, saddle-shaped mushroom perched quietly on a decaying log, don't just see a static object waiting to be categorized. Think about the diagnostic muddy waters it emerged from. Think about the ice it breathes, the toxins it might be hiding, and the genes it might be trading. Ask yourself, what invisible chemical weapons, what silent microscopic wars are shifting beneath your boots right at this very moment?
Nature is always hiding its most profound, terrifying, and beautiful secrets in plain sight.
It really is. Keep your eyes open, keep your curiosity sharp, and always keep questioning what you think you know.